discuss the main steps in the digestion of proteins as the food passes through different parts of the alimentary canals. So students, now let us learn the main steps in the digestion of proteins as it passes through the alimentary canal. So we know that the alimentary canal begins from the mouth and it ends till the anus. So in the mouth there is no digestion of protein. So in the oral cavity, so here we can see the oral cavity, pharynx, esophagus. So in this three region, protein digestion does not take place. So the digestion of protein occurs in the stomach. So once the food enters into the stomach, the initial digestion occurs in the stomach. Partially the protein will be digested. That means it will be broken from a polypeptide into a small peptide. Later the food will be passed into the intestine. In the small intestine, the pancreatic juice and the intestinal juice both are released into the small intestine, and in the intestine the complete digestion of protein takes place and the amino acids will be absorbed by the villi and then it is sent to the liver. So in the liver it gets converted to protein or it will be released into the blood for further transportation. So let us learn about the digestion in the first place, that is in the stomach. Now the stomach contains gastric glands. So here is a gastric gland. Now the gastric gland secretes gastric juice. It contains inactive enzymes, pepsinogen and renin, and along with that it secretes HCl. So in the stomach the initial digestion starts. It contains gastric glands. The gastric gland secretes gastric juice. Now the components of gastric juice are HCl, pepsinogen and renin. Now HCl is a chemical which creates the acidic condition in the stomach. The pH of the stomach is around 2. Now this acidic pH is produced by the HCl. So the gastric glands contains parietal cells. These parietal cells secretes HCl. Once the HCl is secreted, the gastric juice also have certain inactive enzymes, that is pepsinogen. So students, these enzymes, pepsinogen and renin, they are the enzymes required for the breakdown of protein, but generally they are present in the inactive form. So before the digestion, so these enzymes should be activated, and that activation is done by the HCl. So once the food enters into the stomach, it mixes with the acidic gastric juice, and once the food gets mixed with the acidic gastric juice, the pepsinogen which is released by the chief cells gets activated into pepsin, and this activation is done by the HCl. So once the pepsin gets activated, it converts the other inactive enzymes prorenin into renin. So now both the enzymes are activated. So first let us see how pepsin works. So pepsin acts upon protein. Protein is a polypeptide. It breaks the proteins into proteases and peptides. So peptides are nothing but short chain of amino acid which is smaller when compared to the length of a protein. So it breaks the protein into peptides and proteases. Now the role of renin. So we've learned that renin is required for the breakdown of the proteins present in the milk. So generally the proteins present in the milk, it is soluble in nature. Now renin acts upon the proteins present in the milk and makes them insoluble. Now these insoluble proteins will be further acted upon pepsin and they break them into proteases and peptides. So here the proteases and peptides are not the monomers of the protein. The monomers of proteins are amino acid. So we can say that in the stomach, partial digestion of proteins occur. Now this food is moved into the small intestine. Now in the small intestine, the complete digestion of the protein takes place. So the small intestine receives pancreatic juice from the pancreas, and the small intestine itself secretes juice, that is intestinal juice, which is called succus entericus. Now both pancreatic juice and succus entericus, they have enzymes which can digest protein. So let's learn first what enzymes are present in the pancreatic juice. So pancreatic juice have 3 inactive enzymes which is required for the digestion of protein. They are trypsinogen, chymotrypsinogen and carboxypeptidase. So these are the 3 protein digesting enzymes secreted by the pancreatic juice. Now all these 3 enzymes are in the inactive form. So they should be activated before digestion takes place. Now once the food enters into the small intestine, the enterokinase, so this enzyme released by the small intestine, converts trypsinogen into trypsin. Now the one enzyme has got activated, that is trypsin. Once trypsin gets activated, it converts the other 2 enzymes, chymotrypsinogen and procarboxypeptidase, into its active form. So chymotrypsinogen becomes chymotrypsin and procarboxypeptidase become carboxypeptidase. Now all these enzymes are present in an active form. So enterokinase secreted by the intestinal mucosa. This is nothing but the wall of the small intestine. It activates trypsinogen into trypsin. So once trypsin gets activated, the other enzymes chymotrypsinogen and carboxypeptidase also gets activated. Now trypsin converts proteins into peptides. So similarly, this reaction, both chymotrypsin and trypsin, they convert proteins into peptides. So that is a short chain of polypeptides. And the last enzyme, carboxypeptidase, acts upon the peptides and starts removing amino acids from one last end. So carboxypeptidase always identify the carboxyl terminal. So every protein has got 2 terminal, one amino terminal, the other one is carboxy terminal. So if this is a polypeptide chain and this is the carboxy terminal, and if this is the amino terminal, the carboxypeptidase recognize the carboxyl terminal and start removing amino acid from this end. So every time the enzyme acts upon peptide, the peptide becomes shorter in one amino acid. It becomes just shorter in the length by removing one amino acid. Now these dipeptides, so dipeptides means 2 amino acids are connected by a peptide bond. So this is called dipeptide. Now this dipeptide will be released to its simplest form, amino acids, by the enzymes present in the intestinal juice. Now intestinal juice has got varieties of enzymes. It has enzymes for the digestion of proteins, carbohydrates and fats. But here we are only concentrating on digestion of proteins. So the protein digesting enzymes are dipeptidase. So this dipeptidase acts upon dipeptide and break the peptide bond, releasing the amino acids. So hence in the small intestine, the complete digestion of proteins occur. So we learned that the protein digestion begins in the stomach and then it continues in the intestine. So in the stomach, there are pepsin and renins which partially digest the proteins and break them into proteases and peptides. Now these proteases and peptides enter into the small intestine. In the small intestine, it gets break down into amino acid by the action of dipeptidase, and before that the pancreatic enzymes trypsin, chymotrypsin and carboxypeptidase helps break down this proteins into dipeptides.